Speaker of the last session of the day, Lokesh Joshi. And as you probably gathered, Lokesh was one of the pioneer members of the Institute. And Alyssa already told you about the great little group on glycobiology around choline brophy. Very impressive group of people, and who've all gone on to do wonderful things. So Lokesh has a very distinguished career. He's director of the Center for Glycosciences. Oh, he was director of the Center for Glycosciences, but he's wait a minute, where are we? You have an administrator, vice president of research at Galway, as well as the Bristol Myers Squibb professor at the National Center for Biomedical Engineering Science at the National University of Ireland, Galway. So Lokesh does it all. Lokesh, over to you.
Thank you, Stu. Really appreciate it. Really want to thank everybody. Thank you, Stu. Thank you, Josh, for inviting me here. It's great to be back. And it just brings old memories. So for those of you who don't know about Galway or Ireland, I'm based now in the University of Galway, which was founded in 1845. It's an amazing place. So if you haven't been there, you're welcome anytime. And my talk is going to be from Valley of the Sun to the Wild Atlantic Way, which is what the west coast of Ireland is called. So I guess following the same kind of theme, we were asked to talk about our career from biodesign to where we are now. So there are three things that I really learned. There are a lot of things we learned, but there are three things that really stand out to me, what I learned at ASU in biodesign. One was that this was still is a very inspiring place. You know, you meet some incredible people, amazing thinkers, but it's also, you know, so that kind of place and people is very important. Also give us a sense of purpose and develop the vision, what you can do with your knowledge. So with that, when I moved on from here, I went to Ireland. I continued on with my research. And then for the purpose, I wanted to make an impact beyond academia. So I started a company there, which is doing good work. So I'm going to talk about it a little bit there. But the whole idea was that the academics publish papers, have grants, have graduate students. But what are we really doing for the people out there on the streets or just anywhere in the world? And then in terms of the vision, I wanted to bring my training, my learnings from ASU in biodesign to help the university I went to. So I then became the vice president for research and innovation for 8 years of the university, which in fact took me away from my own research. But my whole purpose was to serve the community of researchers in the university across the board, which actually worked out really well. But before I go into that, I really want to say huge thanks, but it's more gratitude to a number of colleagues in ASU in biodesign. Alyssa is here, Elizabeth is here, and Giovanna is here, and a lot of people, but also my mentors. I think one of the top people in the mentors is Charlie Hansen sitting in the back. If it had not been for Charlie, I wouldn't be here. So thank you, Charlie, for all your mentoring and advice to me. Really appreciate it very much. Because I think one of the things for younger people is, I think, it's not just how good you are. You need people who help you, you know, in all different ways, and one needs mentors. And people who have mentors are very fortunate. So I'm really fortunate person in that sense. So in terms of, I have two parts of my talk. One is about research, one is about impact. So in terms of the research, I love sugars. I love eating sugars, and I love researching sugars. So that's really my passion. It's been for a long time, and I just think this is an incredible field. I think it's really people are fortunate who love what they do, right? We are very privileged people that, you know, we work in an environment, you just have ideas, and you can implement them as long as you can get funding. But really, I cannot describe how much I love glycobiology, and it's incredible. So as some of you who were there last night when Caroline Bertozzi gave her talk, and Alyssa spoke about it earlier today, is that all cells are covered with glycans or sugars. That is all cells on this planet, right? Which is incredible. And at the same time, not many people know about it or consider it when they do their research or science. This is the immune cell, and the black band around it is the glycocalyx. And then these are two bacteria. One is pneumococcus, and then a bacteroides. Again, you can see the glycocalyx around bacteria as well. Now, this is a sheep microvessel, and Alyssa showed some elegant slides of the vasculature system, and you can see about 500 nanometer thick glycocalyx. is like the seaweed, that's there, and it affects everything, whether it's atherosclerosis, inflammation, cell rolling, protein uptake, and everything. But also, this glycocalyx is dependent or affected by the stress of all different kinds of disease states. So it's something that even if you were to open the textbook right now, endothelial cell won't have the glycocalyx, it will just be a cell line or a layer of cells. So this is the beauty that we're talking about, which is always there, but it's almost unseen, and people they're happy to ignore it most of the time. So why should we study glycosylation? Why should we study sugars? So there's a long list of things, but on the right-hand side is a cartoon of two cells. One has all the sugars sticking out in different forms, where there's proteoglycans, glycolipids, glycoproteins, and then there are cognate receptor molecules that are called lectins, and that's the language that the cells speak. So it's the molecular language that cells talk to each other using sugars and proteins, and they involve in a large number of, functions, but some of them could be immune system inflammation, cancer stem cells, host microbe interactions, protein biomarkers, and therapeutics. And these
all cells, should there actually be sugars on the cell?
There should be, but I think this is just to simplify it. So what we look at, it's almost like a biological Velcro, right? But it's much more complex than a Velcro we think of, because this is a very dynamic Velcro, and it changes all the time. But there are challenges in the field, and that's why the field has lagged behind the genomics and the proteomics and other omics, is the structural complexity. When we talk about glycobiology, this is a small field of biochemistry, but within glycobiology, people are experts in one thing, whether it's ending glycans, oling glycans, proteoglycans, glycolipids, GPI anchors. But even, I don't know anybody who's in glycobiology who knows about all glycobiology. So we end up becoming, all of us are reductionist, but biology is much more complex. The structural complexity is there. Analytical techniques are still way behind where we are in genomics and proteomics. Informatics, again, is not fully developed, and the high throughput glycomics is not there. But people have been working on this, and hopefully one day we'll be able to make it easier for non-glyco people, because glyco people, they love doing it, right? But it's really everybody else. Can they also do it in their labs? That's the important part. Now, this is an example. We talk about Ig molecules, and at the bottom, there's a circle around Ig1, which is just one of the immunoglobulins. Ig1 itself has more than 36 types of glycosylation states. So when people just measure Ig, that's not good enough. It's the glycosylation state, because the glycosylation of Ig affects its function, right? And in different diseases, it goes up and down. So we're talking about, I always I think that biology is so complex, but people like us or anybody, they reduce it to our own level of understanding, right? So this is really where we are, that it adds another layer of complexity and beauty to our systems. So the way I look at it would be that if this were our cell, and you can look at all these trees around it, we can tell clearly standing here that green is healthy, is getting kind of yellower, then there's fall, and there's kind of winter or just no leaves. And people like myself and a lot of my colleagues in glycobiology think about that. Could we look at the cells and then tell the status of the health of the cell by just looking at the cell from outside without having to go inside the cell? Because what's happening outside is determined by what's happening inside the cell. So that's one thing that we try to look at, is what kind of sugars are present on the cell, and then they're being shed in saliva, in urine, in blood. Can we measure them? And that's the glycobiomarker discovery program. So this was actually a review published just actually maybe 2-3 months ago, and it's one of the most comprehensive review of glycobiomarkers in different cancers. And I don't want to spend too much time, but just to say, there was an emphasis yesterday in cancer and glycobiology that most of us think that, okay, they're protein biomarkers, but they really are either glycoprotein biomarkers or glycans themselves, right? So measuring the protein itself isn't good enough. We need to measure protein and the glycans, or the glycans, in order to be able to basically make the right assessment. So this is, again, it's a huge field. So what I learned in Biodesign Institute is really, in terms of technology, a couple of things. One was these microarrays. So my lab then and now has been printing lectin arrays and glycan arrays. So what we do is that we print there are 50, 60 lectins, They are mostly plant-derived. They're not quite as specific monoclonal antibodies, but they are specific enough, so we print them. And then we also print glycans. So one thing we've done, we've taken the whole human gut glycome and put it on a chip. So then we can put any microbe and just see how they bind to the gut glycome. But we also have other things. So basically, then we can take an antibody, a cancer cell, a stem cell, or a neurocell, and we can label the cells, and we can put them on these arrays and start generating signatures of these cells. So we have used this, and you can see just two different types of signatures there. So basically, you're looking at the handshake between the lectins and the glycans. So you can do both sides, right? So we, in my lab, for the last 15 years or so, have used this for host microbe interactions, both for commensals and pathogens. We've used it for cancer-associated glycomes, primarily for 2 or 3 different cancers. Glycoprotein signature is the big thing because biopharma industry is very interested in making glycoprotein recombinant molecules that have the right type of glycosylation. I'll come back to that in a second. And we've also done some work in stem, immune cell glycome, and biomimic discovery. And the biomimic discovery also we actually learned and developed in Biodesign, and we took it with us to Ireland, is because the lectins are not very specific. So there is a need in this field to make specific binding molecules. So my lab has been developing different technologies, aptamer, phage display, to generate specific molecules against specific epitopes. Then we print them, and we can use them on a chip. This is the biomimic discovery. So again, there are 2 cells, and you have lectins and glycans on both of them, and they're used for virus binding, bacteria binding, parasite toxins. So the whole thing. So the interesting thing, which really fascinates me, is that no 2 cells talk without the involvement of proteins and sugars, right? If we can understand that, then we can either increase the affinity or we can prevent them talking to each other. So you can use them both ways, right? So my lab has been working for the last 15 years or so in generating specific antibody fragments against all of these epitopes. And they can be used for biomarker discovery, and they can be used for different purposes. So that's something we are doing. And just to give you an example of one particular molecule that we made, I don't know how many of you know, and again, yesterday, those who were at the talk, Caroline talked about how sugars determine the blood groups. And you can have 1 or 2 sugars that are different, and sugars, who cares for sugars? But sugars can determine the immune reaction in our body, right? So there is an epitope called alpha-gal, or gal-alpha-1,3-gal, it's a disaccharide epitope, which we don't make. But most other mammals do. It's the old world monkeys, and we don't make it, but all other mammals make it. So when you make a drug in other mammals, and if that drug has that disaccharide epitope, we have an antibody against it, and that leads to hypersensitive response and hyperacute rejection, right? So this was a big case in 2008 or 10, when one of the drugs called cetuximab, which binds to epidermal growth factor receptor, is used for colon cancer. This is a mouse chimeric antibody, and you can see on the top there's a red blobs there. We don't do glycosylation in those red areas. We only do in the lower part, which is the FC region, not in the FAB region. FC region happened to have this alpha-gal epitope, which the companies did not detect because they weren't expecting it. But patients started dying, and that anaphylactic shocks to the level that doctors and nurses stopped giving it to the patients, right? So it was a huge investigation. And what they found is that the patients who were reacting to these drugs actually had been bitten by ticks before. And ticks carry alpha-gal epitope. So patients who had tick bites, mostly in Appalachian mountain range, had developed the IgE response to these drugs, but the doctors and nurses didn't know about that, right? So it became a big deal. So, and again, there's a whole other aspect of the red meat allergy. I don't know how many of you have read about this. There's some people, unbeknownst to themselves, they are allergic to red meat. And that's partly also because red meat also has epitopes of alpha-gal, and that makes it through our bloodstream, and then we have IgE. And then somebody eats a red meat, and within a couple of hours, they would have an anaphylactic shock. And then the last thing is xenograft rejection. That's one of the main reasons why pig organs cannot be transplanted into humans, is because pig cell surfaces are full of alpha-gal epitope. Anyway, so that's a long story. But until now, there is no way to determine the alpha-gal epitope apart from you doing mass spec, and not every lab has mass spec. So my lab developed a single-chain antibody in 2013 or 14, and as you can see in the plot on the right-hand side, it was highly specific to this alpha-gal epitope, and now it's becoming gold standard in many labs. And we are working with labs around the world where they use our antibodies to detect alpha-gal epitope in their samples really all across the world. The other project that we are involved with is on autism spectrum disorder. So this is a $50 million grant from the European Commission, where our lab is involved in two parts. One is to build the biobank, and we have now built the largest biobank in Europe for ASD children, but also to look at the glycobiomarkers. So again, what we are doing is that we have collected fecal samples, although we will not analyze them for glycobiomarkers, but we're analyzing urine markers, saliva, and serum samples for glycobiomarkers. And it's a kind of complex biobank. So we have both the parents, first sibling who's been identified with autism spectrum disorder, and the newly born child. So the whole family is recruited, and we have collected all these samples from them. And it's a very large consortium of 16 different labs. And it's quite exciting to see how we can develop biomarkers at early stages of ASD. But as you know, ASD has several comorbidities. So it's not going to be one biomarker for all types of ASD. It's a huge spectrum, and people have lots of different other health-related complications. So it's going to be an interesting study. Let's see how it goes. The other aspect of the research that we've developed, and again, as Sudhir said earlier, I think data really is the king. And I remember when Charlie Anson and I came here in 2000 or so, there was a whole talk about the gold mines in Arizona in the last century, the century before. But I really think that data is a gold mine. And as Sudhir was saying before, is that there are a lot of people around the world generating a lot of data, but people are not analyzing them. So I felt that our small lab cannot generate all the data, but what we can do is to ask the right questions and see if we can find some answers. And my field being the glycobiology, I'm interested in how glycosylation changes in different diseases and different conditions. So what we have done, and this is just one little slide, or part of the slide, but there's a lot of work behind it. So we've created a database of all the glycoreactions in mammals or in humans, all the glycoenzymes that put the glycans, that remove the glycans, all the proteoglycans, glycolipids, glycoproteins, all the transferases, all the transporters, and we put a database of all of that. Now, what we can do is that we can interrogate that with any tissue, with any condition, as long as we have the omic data. So now we are looking at the multi-omic data, knockout data, mutation data, phenotypic data, functional significance of all of that. That data already exists. And we query that data with just our lens of looking at glyco-related genes. And we've been able to apply that to inflammatory bowel disease. And the whole idea is that could we discover some novel biomarkers or potential biomarkers using this strategy that have been missed by other people. We've applied it to major depressive disorder, autism spectrum disorder, and space microgravity condition. And the first one is muscle atrophy, which in fact Parag was talking about today. And I think as you can see, the interest that I have now is looking at diseases that are not simple to diagnose. There are multiple they're spectrum diseases. There's no one biomarker for these things. I'm interested in, okay, how does body react? How does body change in terms of its glycosylation? Because it's related to the immune response, related to cell communication. And so we're looking at some of these things, and it has led to some really good papers. There are just 3 papers here talking about, this is about the major depressive disorder. We looked at some biomarkers there, inflammatory bowel disease, and the last paper was published in Nature Microgravity just 2 weeks ago, where we looked at muscle atrophy. And in fact, that happened to be, it's not our field of expertise, but what we are really interested in, we are working with Cheryl Nicholson and Jan Barilla here, is in microgravity, how do hosts and pathogens interact? How does host change, and how does a pathogen change? And if pathogens become more virulent or less virulent, if human beings become immunosuppressed, how does it affect their relationship? And the data already exists in the NASA Gene Lab. So what we are trying to do is to analyze all of that expression data to see what's going up, what's going down, to see if we can make some conclusions which can then be validated by experiments. Okay, so there's a lot more going on, but then talking about impact. So I'm always driven, because having lived around the world, different continents, and having worked with some amazing people, I always think that, okay, academic life is good, it's great, but what are we doing for the people? And one thing that drives me is that how can we simplify our translation of science to the general public, right? So there are two things that drive, I guess, my passion. One is the climate change. I think that is the biggest issue we're all facing. Within that, if you kind of bring it down to focus, is kind of the environmental concern. That although, let's say, for example, during COVID period, we saved millions of lives with all the PPEs, all the wonderful things we made, but the thing that I think about is that a lot of them generated a lot of waste, right? So we saved our lives, but we're leaving that waste for the next few generations to come. Right? So why don't we, people in medical technologies, think about developing medical solutions that also reduce the waste impact on the planet, right? So I actually believe, and this is maybe very naive, is that every company should first and foremost be an environmental company and then do whatever they're doing. So one thing I've tried to do, I founded a company called Aquila Bioscience almost a decade ago, and the idea would be that could we generate or develop some technologies that not only helps people, but also has a very, very small carbon footprint. So you make something that's completely biodegradable, but also saves people. All right. So within that, I think we all kind of understand the evolutionary principles. Is that it's not the strongest species that survives, nor the most intelligent, but the most responsive to change. And if you think about humans versus microbes, microbes are single cellular organisms. They can respond much faster, right? They can mutate. Many of them might die, but they can mutate at a much faster rate than we can ever mutate, right? So we are not gonna win the race. They've been here a lot longer than we've been here. They'll be here even once we are gone. So the idea of creating solutions that will kill all the microbes on our body, in our surrounding, is just not gonna work. So what we are trying to do is that how does nature protect us from pathogens all the time, okay? So there are certain pressures, there are the key drivers there. One is that infectious diseases are on the rise. They now account for 1/3 of the deaths globally. And the whole antimicrobial resistance is a growing tsunami, right? I'm sure some of you have read the report from the British Parliament some years ago that they're predicting that by 2050, 10 million people might die of AMR. But now the report was revised actually maybe 2 months ago. There could be up to 40 million people because they're running out of solutions. There's an overuse and misuse of antibiotics and antimicrobial agents. And then the other part is that the current disinfectant solutions, they're harmful to humans and environment. They're destroying the kind of not only our microbiome, but also soil microbiome. And then FDA is coming up with some rulings that's going to restrict the use of a number of disinfectants for over-the-counter usage, only be available through prescription. So some of these things made me think about that, okay, how can we play a role there? And there's a long story behind it, but what drove us is that the current solutions, they are swimming in the kind of the red tank or red ocean, same old chemicals that were discovered in the Civil War or the First, Second World War, and they're mixing and matching, just adding some scent to it and just creating a whole nasty concoction. But there's a need for disruptive solutions, right? And I hope, and I'm sure there are many other people working on that, but our idea in Aquila Bioscience was that how do pathogens attach to cells, right? And then can we use nature's own mechanisms to create sticky membranes or solutions to remove bound pathogens? And can we do it in a sustainable manner? So again, I go back to glycobiology. That's my passion. That's what I understand, that you have a cell, and any pathogen that's binding has to bind to a carbohydrate or a protein. And this is also true for all the infectious diseases. I won't go through the list, but I've highlighted them in yellow. You can see them, so that's true. So that really is the Achilles heel of a pathogen. If it cannot bind, it is not a pathogen, right? So Nature is cleverer than us. I really believe in it. In fact, as I'm growing older, I'm much more humbler. I really believe that nature has been experimenting for a long time. It doesn't use nasty chemicals to keep us safe. We're exposed to things all the time. We are talking to each other, we are inhaling, we are eating all the time, but we don't get sick every day. How does it happen, right? So to keep humans and animals safe from infection, nature has produced a range of molecules and mechanisms that trap and remove pathogens. Mothers produce milk, we produce urine, saliva, tears. All of these are full of wonderful molecules, right? And one of the functions they have is to identify and trap pathogens and expel them from our body. And you can see on the slide that this is how they bind. So Aquila Biosciences developed a nature-inspired biomimic technology that sticks to human pathogens. So in principle, what Aquila has done, I think in the next slide or so, it has made an artificial cell membrane which has natural proteins and glycans that bind to the pathogens and remove them, right? And it's completely biodegradable. So it instantly captures and removes bacteria, viruses, and fungi and biotoxins, uses a biological Velcro mechanism, and is called pathogen capture technology. By the way, the origin of this technology was the defense forces in Europe. They came to us about 12 years ago that they were worried about their soldiers being exposed to anthrax, ricin, all those agents when they are serving someplace else. And they had nothing to protect the soldiers, especially on their skin surfaces. So he said, look, I don't know anything about that. All I know is glycobiology. Let's see if we can apply it, and it seems to work. So we have been funded by many different agencies, and it's working out really well. So the way it works is that we have a membrane or a surface now, which is purely cellulose-based, which is actually polymer of glucose. We do some reactions, we attach proteins and sugars to them, and it comes down, it binds to the pathogens. You can see pathogens have different surface structures, round, triangle, square, all those things. So these are different adhesins of proteins. The membrane comes and binds to them, and you can remove it. And all you're left with is water on the skin surface, right? There's no chemical reaction. It's a mechanical removal. Because what we do actually, we actually kill all the molecules. There's no biological reaction. It's completely based on scaffolds of proteins and sugars. Right? So it's been tested by many different labs, SARS-CoV-2, obviously, influenza that caused the pandemic in 2010-11. A lot of the BSL 3 and 4 work was done in the Czech Republic because we don't have the facility they do. This is a NATO lab on anthrax, both vegetative state and spores. Spores, by the way, are very hard to kill or capture. Clostridium, Francisella, and toxins, hospital-associated infections, as well as some other like Aspergillus, Propionibacterium acnes. So there is a company now that is doing clinical trial on acne. So something that was developed for defense forces is now being used to tell something for acne, and Candida albicans and malassezia as well. So this is being used now to capture and remove pathogens, toxins, allergens from human physical surfaces. Also, there's another application of capturing desired pathogens or agents to enrich them so you can analyze them better. Surveillance in forensic, there is interest in healthcare and consumer care. It's been used by Irish Defense Forces, Czech Army. This was me. I'm a faculty at the NATO College in Germany as well. And we recently heard the news that we've been given, I guess, a grant to develop this for the astronauts' protection on the International Space Station. So with that, coming back to the whole story, that what I learned here is, and again, I was just so lucky to be surrounded by people who really were big thinkers, inspiring people, that you're lucky to be here really in some ways. And just find your purpose, find your vision, and just stay inspired. You find a lot of challenges, but it's really, as a scientist, we're very lucky to be where we are. And also, I think we need to be the change that we want to see in the world. We can't expect other people to do it. So you have to have that fire in your belly that you want to make a difference and bring a change. With that, I would like to thank you all. This is Cliffs of Moher, west coast of Ireland, about an hour drive from Galway. It's a beautiful place, and I welcome you all to come there. Thank you. And again, just to say, yeah.